It's my pleasure to introduce my colleague from the Department of Biological Sciences, who is also one of our newest COVI fellows that joined us last year, Dr. Jeffrey Stuart. Jeff did his PhD in zoology at the University of Guelph, which is just down the road from us. And then he followed up by doing his postdoctoral work at the MRC Dunn Human Nutrition Unit at Cambridge University in the UK, and then continued with additional postdoctoral work at the National Institute of Aging at NIH in the US. He joined us here at Brock in 2004 as a member of our Department of Biological Sciences. He's published over 40 papers and chapters on the roles of free radical metabolism and DNA damage in the aging process, and has recently, just this last year, completed a book on methods involved in this area of research. So while studying genes that influence lifespan in mammals and dietary molecules that alter gene expression, this led Jeff to become interested in the biological activities of resveratrol, one of those wonderful polyphenolic compounds that we find in wine. And this brought him to the COVI team, and he joined us as a COVI fellow last year. So today he's going to share with us some of his findings on this biological activity role of resveratrol that he's been unraveling with one of his grad students over the past couple of years here.
Thanks very much, Debbie, and my pleasure to be here. I'm going to start today, so I'm Jeff Stuart, Biological Sciences, and I'm going to start today just by giving a brief background overview of basically what the research program that I'm undertaking in my lab, because a lot of it's not directly involved in wine chemistry, wine science by any means. It really is this one molecule, resveratrol, and some related molecules, other polyphenols that are also phytoestrogens that we're interested in. Of course, those are really relevant to the wine industry because I think basically there is money to be made in those. There are some very tangible, positive health outcomes associated with resveratrol. And in fact, that's what I'll talk about today is our effort has been primarily focused on finding out exactly mechanistically what resveratrol is doing in its interaction with human cells. So, just a brief introduction. I came to Brock from the Institute of Aging in the United States, and so I had this real interest in not just aging necessarily, but in what underlies longevity, lifespans. And one observation that we can make easily, if we just take a moment, is that there are there is an incredibly broad range of lifespans just amongst animals. So if we look at the shortest lived animals, which tend to be insect species, we have lifespans of a few days. And then all the way up the spectrum to some of the longest lived species, which are more than a century, sometimes more than 2 centuries. And we basically are just asking why and how this occurs. It kind of shakes out like this, where we see there's actually a really good relationship between the adult mass of an animal and how long they're going to live. So that's a statistically significant correlation. We understand the ecology of why that would happen. Becoming big allows animals to escape predation. But we really don't have a very good idea of how it happens. Why? Makes sense. They're going to escape predation. They invest more energy in becoming larger. That takes time. They have to live longer. They postpone that first reproduction, and everything changes. But really, we want to know, how do they do that? So that involves some understanding of what aging actually is and how it can be postponed. In this case, we spend a lot of time thinking about how the evolution of the aging process is postponed. So these are our 2 main questions, why and how. Pretty broad questions. And we're really particularly focused on the how, because that's the mechanistic thing. Now, unlike Steven Seagal, unfortunately, the cells of longer-lived animals are hard to kill. So this comes into the how category. One thing that comes up again and again when we look at at long-lived species or long-lived strains. These could be mutant strains of mice. what we see is that if we look at the cellular level, the individual cells that comprise that whole body, they're hard to kill. And we measure this in the lab using a variety of assays that are basically just, we throw nasty things at those cells, reactive oxygen, heavy metals, heat, light, radiation, all kinds of things that would kill any biological material. And what we see is the longest lived species have the most stress-resistant cells. So we can plot this on a graph like this, where if we take the lifespan of any individual species and we compare it to a lethal dose, this is an LD90, a lethal dose 90, of any particular stressor or toxin, we can find a relationship like this. Now, a lethal dose is simply you have 100 cells sitting in a dish, you hit them with increasing concentrations of a toxin, and the point at which 90% are no longer viable, that's your LD90, so that's what's represented here. These are individual species, these data points. And what we see is the longer lived the species is that the cells are taken from, the more of any particular toxin is required to kill those cells. Hence, they're hard to kill. We think this is important because we think that a lot of the aging phenotype that manifests in any animal is actually related to the gradual loss of cells due to their death. And that is in turn related to just the kinds of stresses any body, and therefore the cells that comprise the essential organs, are encountering on a day-to-day basis. Now, we can see the same kind of thing if we just look at some strange mutants. A lot of this kind of research is done with mice. They're easy to house. They're a mammal that's very similar, that has a very similar genome and gene expression to people. So they're a reasonable model of human physiology, including cell physiology. And we have a lot of tools with mice. In particular, there's about now maybe 10 strains of mice that are long-lived. And they become long-lived by actually, these are single gene mutations that confer that longevity. So they're exactly the same, these mice, as their brothers or sisters, but they have inherited a mutation in one gene, and somehow that's making them long-lived. Now again, we know sometimes what the gene does, but normally the genes are transcription factors, so they're interacting with a lot of things downstream, and the challenge is to understand what's going on downstream. But in any case, we have a collection of some of these mice, and what we do know about them is that they conform to our rule that we've observed in other instances, where their cells are also more resistant to stressors. They're also hard to kill. So this is a normal mouse. Well, here's a normal mouse, and there is a sister that is called a Snell dwarf. It's named after the discoverer. And the dwarfs, in addition to being really tiny, they're about 1/3 of normal size and incredibly cute. This guy is about 7 grams. They live a long time. They have problems staying warm, which tiny mammals always do. But if you keep them warm, they have a survivorship curve that looks like this. This curve is just, you go back to a population of 100 mice, and at any given time, how many are still alive? They're under ideal conditions in a cage. You feed them what they want. There's no disease. They're in a sterile, filtered environment. And so this would be the survivorship curve of normal mice. This is what the Snell dwarf mice look like. You can kind of infer from this survivorship curve, by the way, that they're not really unhealthy. It's not just that they're living a long time, but they're really decrepit. They really don't start to age. It's the beginning of the aging process that's postponed. So up here, that plateau where there's basically no loss of animals is extended quite a bit, you can see. So that's what the survivorship curves look like. If you isolate cells from these guys, you'll see that the dwarf cells, again, are resistant to a variety of stresses. Now here I've just shown one. This is hydrogen peroxide, but I could have shown you heavy metals, I could have shown you UV light, any of a variety of things. And this is a fairly robust, there are occasional exceptions to these rules, of course, but generally quite a robust rule. So we call what we do now species omics, just because everyone likes to use the term omics, because I think it makes things sound important. And I mean, genomics, proteomics, these kinds of things, the approach is to try to look at all of the, for example, proteomics. You're looking at all the proteins that a cell has produced under a given set of conditions. And sometimes what you might do is something like ask the question, well, what about a cancerous cell? What would its proteome look like compared to a normal cell? And there'd be some information there because a different set of proteins would be made, and you could start to ask questions about what's going on in that cancerous cell. Now, I say speciesomics, and I'm really just joking, but we take a different approach, which is to, there are long-standing theories about what causes aging and what might influence longevity. And our starting point is to pick out the activities that are thought to be important there, antioxidant enzymes, DNA repair activities, things that promote protein homeostasis. These are all components of what's called the stress proteome, the cellular stress proteome. So we know a priori what those things are, and what we do is we go and ask, are the animals using them? So in the longest lived species, do we see a greater capacity to repair DNA? Do we see more antioxidant activities and so on? So that's speciesomics. And we do these, we use two basic models. I'll call them models. And the one is we use Snell dwarf mice that are long-lived. Maximum mice lifespan about 50% longer than normal mice. There's actually a contest on right now, the Methuselah Prize, which is worth about $1 million, a lot of money, for the longest-lived mouse that anyone can produce. And it is actually right now the winner, pending, is a Snell dwarf mouse. We also use a comparative model where we have sampled from many, many species. We have a tissue bank in our -80 freezer, and we continually go back to that bank and consult it to ask these kinds of questions. And then most of those samples are actually just obtained from collaborators, animals that are going to be sacrificed anyway. A lot of them are actually from abattoirs locally. And all we do is go in and take little bits of liver, brain, and heart. Those are our three main tissues that we use. And those are intentionally because those are highly oxidative tissues. Brain and heart in particular, heart attack and stroke are big causes of death and therefore limiters of lifespan. So we want to know what's happening in those tissues. We don't confine all of our questions to those tissues, you'll see, but a lot of them. Okay, so we've been taking that approach for a while now, I would say 2 or 3 years, and we're beginning to feel like Belarus in the Winter Olympics. I hope no one here is from Belarus. But basically, if we ask, what have we learned? I think we've learned a lot, but mainly we've been learning through negative results. So there's all kinds of antioxidant enzymes, for example, that have been implicated in lifespan. Most of those, we don't see much going on. So you can be a Snell dwarf mouse and longer lived. Doesn't matter, you don't have necessarily higher activities of all of these antioxidant enzymes. You can be the same thing, and you may not have higher levels of proteins that confer resistance against proteotoxic stress, all kinds of things like that. We see a lot. We've identified a lot of things that you would expect, theoretically, should be coming out higher in the longer-lived species or strains of mice, and they don't. And we have basically one gold medal here, and that gold medal belongs to an enzyme called manganese superoxide dismutase that nobody's heard of, but it's an enzyme that's actually found in mitochondria. And mitochondria are organelles that are found in all of your cells that are involved in metabolism. They're the things that actually take the energy that you eat and convert it to energy that you can do stuff with, like move muscles, make action potentials in brain and make your liver work and so on. And there's been a long-standing theory that actually attributes aging and longevity to something going on in mitochondria, and that's something having something to do with reactive oxygen metabolism. So as you metabolize, your metabolism is oxidative, that's why you're breathing in all that oxygen, and a necessary, seems unavoidable byproduct of that is the production of first superoxide, which is a form of oxygen that has an extra unpaired electron, and then downstream other so-called reactive oxygen species, or ROS for short. This takes place in mitochondria. This enzyme is directly involved in that. So the very first reactive oxygen species, superoxide, is the substrate of manganese superoxide dismutase, or manganese SOD. And so this initiates the detoxification or neutralization reactions. So this at least has a sound theory framework that it can fit into. But it is interesting that when we looked at there's all kinds of, this is only one of maybe 10 antioxidant enzymes, and it is the only one that seems to be showing up as a correlate of longevity. So it brings things into the mitochondrial realm. So these little organelles that actually have a very important role and have long been implicated in aging and lifespan. So, by the way, this is in dermal fibroblasts. Dermal fibroblasts are easy to work with. They're human cells. These come from human lungs. And you can grow them readily in the lab. So they're great to get started with and do lots of experiments. You can freeze them down and thaw them out and then work on them again. And that's a nice result for fibroblasts, but ultimately you want to be, you want to know, not all cells are alike, not all cell types are alike, so you want to go beyond fibroblasts, which we've done. And we found when we looked in mammalian brain tissue, and here I've actually put the species names on, we've got quite a range, and we've actually got more than this now, and this result holds up. What we find is that manganese sod again, so here we actually had 15 different correlations we did, and this was only one of 2 that were actually significant. And again, it's manganese sod, and manganese sod specifically in brain, that is a positive correlate of longevity. So, I mean, we want to know what's the significance of manganese sod, what's the significance of that? And I would argue that based on everything we know about how mitochondria work and the role of that enzyme, the significance lies here, that reactive oxygen species, or ROS, that are produced during metabolism. One thing that they do, so that's normal actually, it's normal to have some of those forms of oxygen produced, but the overproduction of those is associated with the peroxidation of the membranes that comprise the outer boundary of mitochondria, and that's a chain reaction. So once peroxidation begins, it can actually cause big sheets of membrane lipids to become peroxidized, which is just generally bad. Things will not function normally under those circumstances. And once that happens too, this is associated with the release of cytochrome C and the initiation of apoptosis. So what does that mean? Cytochrome C is just a protein that mitochondria have. It's found nowhere else in the cell, just in mitochondria. And when it's in mitochondria, everything is good. It's actually playing an important role in energy transduction, making ATP from the stuff you eat. But what happens is it can actually be released from mitochondria. So here's cytochrome C. This is a mitochondrion in cross-section. And here you can see cytochrome C in the mitochondria. And this is some that's sitting outside. This is a sort of abnormal situation. And it occurs immediately preceding cell death. So this is actually cytochrome C, we now know, is in fact a cell death signal. And this is shown here. I'll just show you quickly. This is cytochrome C that's actually causing the formation of an apoptosome. Now, what does that mean? One way that cells die, and it's the best way if you're going to have cells die, it's the best way for that to happen, is called apoptotic cell death. It's a controlled and regulated form of cell death where there's not, the neighboring cells are not affected. That's a very bad thing. Necrotic cell death, where it's not controlled and cell contents spill out and affect all of the neighboring cells, is terrible. And that's what you would have in heart attack and stroke often. You have both kinds, but lots of necrosis, that second kind. So this is a more regulated kind of cell death, but in the end, it's still cell death. It's still the loss of cells and then the potential for actually degeneration of tissue. And again, a lot of aging disorders are associated with tissue degeneration that can be traced to cell death. So anything that prevents cytochrome C release is going to be positive in that respect. And manganese sod does that. It actually, that enzyme that we saw, it actually prevents cytochrome C release. Okay, so that was the background of what we do in the lab, and it was Ellen Robb, a PhD candidate, who came maybe 2.5 years ago now, as an undergraduate student, maybe that's longer than that, to start to work in the lab, and we thought about what she might want to do for her undergraduate thesis, and we came up with resveratrol. And the reason for that was that resveratrol at that time was in the news. It had been for a while, but it was becoming more interesting to me because some direct connections were made between resveratrol and lifespan and longevity. Now, cardiovascular effects have been known about for a long time, some anti-cancer effects as well, but specifically in the early 2000s, there were some links between resveratrol and actual lifespan began to be shown in the literature. So, now a lot of this is hype, and it is a challenge to decide which is which. But certainly, there was all kinds of, in the public press, there was all kinds of stuff about, all the wonderful things that resveratrol could do, and there began to appear all kinds of resveratrol nutritional supplements. I assume the Twinkie thing might be a joke, but people were experimenting with the idea of giving resveratrol in lots of different dietary vehicles. And certainly there is a big literature on anti-cancer activity, resveratrol's antioxidant activity, aging, and neuroprotection. And those have all been developing for a while. There's certainly believable evidence for roles in these things. But again, hype. It's an order of magnitude less than maybe is professed sometimes. So the starting point for us then and for Ellen was to, if people are going to start taking these as nutritional supplements, resveratrol, then the question really becomes, what is the long-term chronic effect of resveratrol on human cells? So we picked some of our favorite human cells. These are the lung fibroblasts again, because we knew a little bit about these guys already. And we asked, what happens? What is the effect of exposing those cells to resveratrol? Not just for a short period of time, which is basically the experiment that everyone had done up until Ellen, a few hours. And also, up until Ellen's work, people had assumed that resveratrol has some marginal antioxidant activity itself. And so the thought was you put resveratrol in, its chemical antioxidant activity is beneficial, and you protect cells. Now, it turns out that if you put resveratrol in, incubate cells with it for a while, and pull it out and get rid of resveratrol completely, there's still an antioxidant, a very strong antioxidant effect. So that's not real. And it was basically, that discovery was made because Ellen exposed those cells for up to 2 weeks. And it degrades in the medium, so that means every day going back and replacing the medium with new, fresh resveratrol. And what we did was, because we were interested in what was going on, again, we asked the same kinds of questions that we've been asking in other contexts. And we asked about a whole suite of antioxidant enzymes. Again, there's a good 10 of them that are known to have some fairly important roles. But we found that despite that fact, most of them were not at all affected by resveratrol. So this is just sort of a representative graph which shows you nothing. Nothing going on. You see resveratrol. These cells are given resveratrol for up to 2 weeks, 24 hours, 72 hours, or as long as 2 weeks, continual exposure. And this is a cytosolic superoxide dismutase. So it's the brother of that mitochondrial one. But here now it's just in the cytosol of the cell, has no function in mitochondria. And we see that there's no effect. We repeated that kind of measurement with lots of other enzymes, antioxidant enzymes, and we saw nothing. But what we saw with our manganese sod again, this mitochondrial enzyme, was that the levels just went through the roof. So this is a Western blot. We're measuring the levels of the protein itself. Now, we also measured activity, and it corresponds very nicely to this. But what this is telling us is that more manganese sod, more of that protein is being made, and a lot more. So that is about 6 times. By 2 weeks, it's a progressive, ongoing thing. It does seem to plateau after a period of time, but at 2 weeks, it's becoming close to the maximal amount, and it's a 6-fold increase in the level of that important enzyme. Now, that could have been just a lung fibroblast story that was somewhat random, and it would have not been very interesting if that was the case. So one of the first things that we did was to actually look at other types of cells. Excuse me. Myoblasts are a muscle progenitor cell. So in your skeletal muscles, you have lots of little myoblasts. They're a satellite cell. They're a tissue-resident stem cell that is there in case you lose some muscle cell from injury. And these things come in and they differentiate into muscle cells and replace what's lost. It's not a perfect thing, but you have some potential there to do that. And what we found was if we did the same kind of experiment with our myoblasts, we also saw an induction of manganese sod with resveratrol. So we knew that this was true for fibroblasts and for these myoblasts. And then we looked at neuroblastoma cells. And this is like it's very difficult to actually culture adult mammalian neurons in the lab. But so we have some other neuron-like cells, and these are derived from neurons. But they're slightly different. But in any case, we can also show that same effect with these neuroblastoma cells, which are human. So we conclude from that it's actually a more universal effect. Now, we would hypothesize that being the case, that resveratrol is actually inducing manganese sod expression. And we know what we want to know is, so let's take away resveratrol and see if what it does. We think it's going to increase cellular stress resistance because that's the way we think all of this works. And this time we're right. So this is some data, and this is hydrogen peroxide exposure of a bunch of different cell types. And what we have here is control, which are cells not exposed to resveratrol, and then cells exposed to resveratrol. And these are all the same cell types that I've mentioned. These MRC5s, I probably could have labeled them differently for you. These are the lung fibroblasts, so you did see these already. This is how resistant to stress they are normally. And then after a period of time of exposure to resveratrol, I think in this case it's 72 hours. And again, resveratrol has been withdrawn at the time of the exposure to these chemical toxins, so it's not a direct resveratrol effect. We think this is the manganese sod effect. These are the myoblasts, C2, C12, that's kind of like their trade name, the same effect, and these are the neuroblastomas, the same effect. And this is simply a different stressor. So this is a result we see for a broad range of stressors. So yes, we see increased manganese sod expression, and we see increased stress resistance. Now, the other thing that we immediately see when we expose our cells to resveratrol in the lab is that they stop growing as quickly. So we call that population doubling time. And the longer the population doubling time is, then the longer they're taking to grow. They grow by mitosis. They double, and then each of the daughter cells double, and so on. It's an exponential thing. When we add resveratrol, that growth rate slows down. That's also important because there is a strong link between resveratrol and cancer prevention, including slowing the growth of tumors. And this is a very robust result we see with many different cell types, not just cancerous cells, but also normal cells. And we know quite a bit about how that's going on. It's actually a signaling phenomenon where inside of cells, events are signaled by a variety of activities, especially that have to do with oxygen. And so this manganese sod effect is working through that way. I'm not going to talk about it much, but we know that's what's going on. So one of the next things we wanted to know is just how. I mean, we always like to know how. And we want to understand, we throw resveratrol in there, things change in a way that we appreciate. But we'd like to know exactly what is resveratrol doing. And so we consult the literature. And one thing that's been in the literature for quite a while, a nice paper in 1997, which I haven't cited, but talking about resveratrol being a phytoestrogen. It's funny that it got left in the dust a little bit as people started to think about resveratrol doing other things. But as you'll see, in our case, it's working this way as a phytoestrogen. In other words, resveratrol has structural similarity to estrogens, like estradiol. That's a specific form of estrogen. Structural similarity, not tons, but they're both phenolic. And in any case, it it doesn't matter that much how structurally similar they are. What matters is the experiment where you actually take the estrogen receptor and incubate it with either of these shows us that, yes, it does actually bind that receptor productively. And that's how hormones work. They bind a receptor and cause something to happen. So when I say it binds it productively, I mean resveratrol binds the estrogen receptor, and the things that would normally happen with estrogen binding the estrogen receptor are indeed happening. So estradiol, so it's a phytoestrogen, we think, and so we just ask the question, the things that we see with respect to manganese sod, are they going through the estrogen receptor? And to understand that, we started here. First of all, we would want to reproduce that result with estrogen. In other words, instead of exposing the cells to resveratrol, we expose them to estrogen, and we want to see that it's also causing manganese sod to be upregulated. And we had some reason to think that would happen, and yes, indeed it did. DMSO here, dimethyl sulfoxide, is just a vehicle. It's just something we add the resveratrol in, so it's like nothing. That's why it's empty. Here's our 50 micromolar resveratrol, which is a very small amount, but here's even less estradiol. And we see that estradiol is quite good at inducing manganese sod expression, so we can confirm that. Now, this is also not surprising that this is about 500-fold less estradiol than resveratrol. We also knew that, and not surprisingly, the estrogen receptor has a greater affinity for estradiol than it does for resveratrol. Again, that's as it should be, really, but it just means that you're going to have higher concentrations of resveratrol to get that receptor going. Now, you would also want to do the experiment where you inhibited that estrogen receptor and saw your effect go away. And so that experiment was done. This is control again, just the vehicle. In this case, resveratrol was added to the cells again, but also the inhibitor of the estrogen receptor specifically. And we see that there's no effect. There's no ability now of resveratrol to actually induce manganese sod expression. And the same thing for estradiol, as we would expect. If we inhibit the receptor for that hormone, it's not going to be able to perform its function. So again, we're zeroing in on this is how that's happening. Now, there's an estrogen. Actually, unfortunately, estrogen receptors are, it would be nice if there was just an estrogen receptor, but there are estrogen receptors in the nucleus, in the cytosol, and in the plasma membrane of cells. And there are also two broad categories of estrogen receptors. There's an alpha isoform and a beta isoform, and they have different downstream effects. So, in addition to knowing that it's an estrogen receptor interaction, you really need to know which isoform. And the way we've done this right now, there aren't there's not great inhibitors of specific isoforms. They're too similar. But there are some very good agonists or stimulators of specific isoforms. And these two compounds, DPN and PPT, are specifically going to interact with either the alpha or the beta isoform of the estrogen receptor and elicit some kind of response. And what this is showing us is, again, here we're measuring the manganese sod induction. And here's control. This is the alpha receptor, and we see nothing. And here's the beta receptor, and we see something quite strong. So we think that it's actually the beta receptor that is responsible for the downstream signaling event that causes more manganese sod to be made. So yes, we have this working model now where resveratrol interacts with cells, a wide variety of cells, not just, and these are human cells. And it's in the low micromolar range, interacts with these estrogen receptors, and this would be beta, we think, primarily or even exclusively. And I haven't talked at all about these secondary steps, but they end up in the nucleus where they're causing the transcription of this, in particular, this one gene. And I've left out, there are certainly other effects, but this is a very important one, we think. Manganese sod, which is produced and then goes into mitochondria where it has these effects in terms of preventing apoptosis, and it alters reactive oxygen metabolism in a way that is actually influencing cell growth. So that's all great when you do those kinds of experiments in dishes, cells in dishes in the lab. You don't have stomachs, small intestines. He may not either. you don't have those kinds of things interfering with the chemistry of what's going on. You add resveratrol, and if you're refreshing it in the medium every day, you can actually do the experiment where you show that it's still around. We know that the highest dietary, when something's excluding supplemented things, dietarily you're going to come across the highest concentrations of resveratrol in a usable form, probably in red wine, and a few other select items in the diet. So you have the choice of either drinking vast quantities of red wine to get resveratrol levels high, or find some way around that, or maybe a combination of the two. So the problem here, though, is resveratrol bioavailability. You ingest, just because something has an activity in the body doesn't mean that it's going to get past the digestive system, whose specialty is destroying stuff, breaking it down into component parts, modifying it, making it inactive metabolites. I mean, that's a protective function. It's a good thing. But in terms of something like resveratrol with some beneficial effects, it's going to interfere with your ability to do anything with it. So what other people have found is that dietary resveratrol intake, the amount taken in versus the amount that's free flowing in blood plasma is very, lots taken in, very, very low in blood. So we knew we needed something in the micromolar range, and we started to experiment with ways to get that. And we've done some work with John Bracci at the University of Toronto, the University Health Network, experimenting with different ways to actually introduce resveratrol. And of course, we use mice because it can't really be done with people. And we've tried a few different ways of getting it in. We just added it directly to their diet, their normal mouse pellets, the stuff that they eat. And we really didn't, and our downstream, what we're looking at downstream is the ability for brain manganese sod levels to be induced. We want to show that that's actually being turned on. So that's what we're measuring here again. But nothing with the standard diet. We've added a ton of resveratrol, really a mega dose, not toxic by any means, but lots of resveratrol, and we see nothing. We tried another way to do this, a mini pump, which is actually just, it's surgically implanted. It sits just underneath the skin, and over 30 days, it releases every day small little package of resveratrol in the vehicle. But again, we didn't see anything with that. And when we introduced resveratrol in a high-fat diet that was safflower oil, that's the fat source, we saw that it was actually able to elicit this effect. Now, other people have shown subsequently similar kinds of things. We know for sure that the way it's actually introduced into the body, into the digestive system, is important. And exactly what's going on there is another question. But this illustrates nicely. These are all the same amount of resveratrol and very different downstream effects. But of course, most importantly, we've shown that we can actually get this effect. We started to work with Paul Zelisko here at Brock because he works with silicon and using silicon to encapsulate things, chemicals, molecules that would normally get broken down in the digestive system and that you want to get past where most of that happens, stomach and small intestine. And encapsulating things in silicon is pretty good for that. It protects. And then normally, those contents can be released in the large intestine and absorbed. So we tried that. And this is not statistically significant. This is the one. So these two are controls. So this is nothing. This is just the silicon with no resveratrol. That's the silicon with resveratrol. And it looks like a hint of something, but it's not. It's not real. It's not statistically significant. But it's enough to make us think that we want to go back and do some more work with that. And so that's what's happening is experimentation with different formulations of silicon that affect the release kinetics of the contents, resveratrol, differently. So we'll be doing that this summer, I guess. So conclusions, and this is an ongoing thing, but this is what we know primarily from our own work, but certainly there's a lot of work being done in this area right now. We think that it's interacting with resveratrol in human cells, interacts with the estrogen receptor beta isoform specifically, and it elicits a transcriptional upregulation of that manganese sod, very significant one. And it's actually different in different cell types, but it can be as large as almost an order of magnitude, almost a tenfold in some cell types. We know that that's associated with increased stress resistance of cells, and we're doing the experiments now to look at cytochrome C release in mitochondria, from mitochondria. And we know that this is really more from other people's work, we know that it directly does alter mitochondrial ROS metabolism, and from our work we know that that is influencing, it's actually inhibiting cellular replicative growth, which is what cancer cells do, so it matches up there nicely. And we know that, that although it's a pretty low amount of resveratrol that you could get in with most normal red wines, you can actually, there are more effective ways to introduce resveratrol. And that seems to be associated with high fat. And that's not to say that that's the best way. Of a few ways we've tried, it's the best, but it must just be the beginning of maximizing the dietary introduction. And then maybe silicon nanocapsules are one way, but many people are working on ways to increase that bioavailability. So that's what we know. And where we're going with all of this is we have been, we are interested certainly in more work in this area to increase resveratrol bioavailability. And one thing we've thought about is supplementing red wines with resveratrol because it has no major negative effect, even at fairly high levels, on taste. It's certainly not toxic. And it may be that that kind of red wine matrix is able to support the stability of resveratrol and get it into the system readily. Especially you have all these polyphenols that complex with each other, and that may offer some protection in the gut. But we're also exploring other polyphenols and other phytoestrogens. There are other dietary phytoestrogens found in soybean, in addition to red wines and lots of other places. So we did some work with quercetin, which does not do any of this, we know that. But the soybean molecule, geneticin, does do some of these same things. And some work with the metabolites of resveratrol, because those may be, what I'm talking about now is metabolites that the gut creates, modified versions of the molecule that do make it into the system that may still have biological activity. We don't know much about those. And then, of course, human trials eventually, knowing I'd like to be able to match up manganese solid levels not in brain of people, but even if we knew something about some of the white blood cells that we could take out of blood of humans, that would be interesting, because there's an immune story there as well. So this work has mainly been supported by NSERC and the infrastructure by the Ontario government and Canada's CFI. And Ellen is supported by NSERC's Vanier Award, which is a very generous scholarship for graduate students. And thank you very much.
We've got time for questions from the audience.
Yes?
Did you try to see the biological activity of resveratrol with polyphenol as a synergistic effect?
The bioavailability
Of resveratrol with polyphenol as a synergistic effect.
resveratrol with other polyphenols? Yeah, we haven't done that, but that's, we would like to do the experiment where we take red wine that's full of polyphenols, and there's certainly substantial quantities of polyphenols, and then add a ton of resveratrol to that red wine. And Gary Pickering has done those experiments. You and Gary Pickering have done those experiments. But they know that it's stable on the shelf and that it doesn't have a terrible effect on taste. And then what we'd like to know is, what does it do when it's ingested by mice? And we have a standing, we have to get that past the animal care committee to be able to feed red wine to mice. But that's, that's the direction that we're going. Vince?
Have any people tried to estimate whether Asians have higher levels of peroxide, this manganese peroxide dismutase compared to North Americans or Europeans, for example, because you did mention that the soy, well, typical isoflavones is another weak estrogen, as far as I know. Would they characteristically have higher levels of that particular activity compared to people who don't have a diet high in those compounds?
Right. So, I mean, the expectation based on that is that if the levels of intake are high enough to sustain, steady state levels of all of those phytoestrogens in appreciable enough quantities to get an effect, then yes, you should, because we see those effects in all mammals, including people. We see them in a wide variety of cell types. So it becomes a question of, is there enough?
But nobody's made those measurements yet in a...
I have to know the measurements have not been made. No, they'd have to be made. They'd have to be made on, for example, brains of, I suppose, cadavers of people from those, and that's really tough to do.
Yeah, you can't get mitochondria from people.
People really don't like that. No. I haven't even tried.
Did they not show at one point that phytoestrogens, like even in soybeans, were working almost contradicting the anti-cancer component at all? Is that almost a cancerous?
Oh, well, they can be. I mean, estrogen
So, I mean,
itself.
if we're saying that resveratrol is supposed to be very good, okay? How do we know if it's going to be a chronic?
Yeah. Well, it's like estrogen replacement therapy.
That's basically
right?
what it is.
Yeah.
So it's becoming almost like a substitute estrogen.
So anything that's linked to the estrogen receptors is going to be potentially affected by something like that. And you can have the same, estrogen, the same hormone, estrogen receptors, the same ones, but they're linked to different signaling pathways in different types of cells, and therefore they elicit different downstream responses. And so you could get some responses that are going to be quite positive and beneficial, and yes, you could have some that are negative. So that's always, when you get into something as complicated as the entire human body, the reason to get away from the cells and culture, then you start to have questions like that. So anything is possible, and there may be an effective range, or it may be that it's good for some people and not other people, depending on risk factors. That's, that there is hype to all the resveratrol and phytoestrogen stuff, which I think is why you need to understand. It's important to understand mechanistically what it's actually doing. Otherwise, you're just trying to match taking up a lot of it with a whole bunch of effects downstream, that some of which are good and some of which might be bad.
What's the main source of resveratrol? I mean, where you can get this the cheapest price?
Oh, I think people are making it now. That there's a substantial amount of it in grape skin, which is why it shows up in red wine as the ethanol form. It's not very water soluble, but it's soluble in ethanol. And so red wine is fermented, I guess, on the skin, and it's actually extracted during the fermentation. So the extracts are coming, some of them from grape skin. A lot of them are sold as resveratrol, but they're probably 2% resveratrol, and the rest.
So for the chemicals that you use for your experiment, where you get
Sigma.
Sigma.
I mean, same old.
Synthesized or?
They're purified.
Purified from the extractions.
They're purified from vegetable extracts. Whether there's other vegetables you can find resveratrol in. So whether that's, I can't remember if that's grape skin.
So we can make the Japanese knotweed.
Pardon?
I think it's from the Japanese knotweed.
I think you're right. You mentioned that. So they're purified from plants.
I guess a related question. The reason I'm asking this is, if it's very hard to get a high amount of this one compound, then because there are other similar chemicals that have similar effects, so we may want to look at some other chemicals instead of this one.
Yeah, and that's one of the things that we're doing, is to start down that road. Other polyphenols and phytoestrogens. To be honest, the story that I told you, parts of it parallel the geneticin story. The soybean, a molecule that's found in soybean, also a phytoestrogen. And so as we were doing this work, we actually went to a conference on phytoestrogens and human health, and we saw all of these geneticist talks, and we realized, you know what, it's actually these two molecules are giving the same basic effects. So where there's two, there's probably lots more. And certainly quercetin has been suggested. We've done the quercetin experiments, and it's not doing this with manganese sulfate. I'm sure there's lots of other molecules, plant molecules, that can do this. Some may be better than resveratrol.
It might be useful to do like a comprehensive assessment of different chemicals. They may have slightly different side effects.
Yeah, and that's one thing on the list.
If you do like a gene expression assay, then you can look at all other effects perhaps.
Yeah, also on the list. But those, some of those are just expensive to do. And so it needs, this is being done right now on my answer grid. And that, you're talking about now an amount of work that can't be done that way.
But gene expression is not too expensive. Like
if you're set up to do that, but if,
$9
yeah, but there is not that amount of money extra after everything else that my lab does has been done. So it requires its own funding stream, as opposed to being piggybacked on another funding stream as it is presently. Ian, you were going to ask something?
Yeah, there was a study done quite a number of years ago on tri-substituted phenols, which are used in packaging materials as antioxidants. You find in cereal packages and things like that. So you've basically got a hindered phenol. So when it gets oxidized, it doesn't form, you don't get phenol oxidative coupling. So you get these stable free radicals formed. Are you familiar with that literature? Because I think there was a time when they fed it to rats and found they lived longer as a consequence of being fed that material.
I'm not familiar with that now.
I'll see if I can.
Sure.
Dredging up memories.
Yeah.
What about sugar substituted resveratrols? What have you learned from the literature? I know that they're not going to be looking, they won't have any weak estrogenic activity, but will those types of compounds be incorporated more, will they go down the intestinal tract and not be degraded as badly as resveratrol, and will that increase, because then you can obviously remove the sugar, and once it's in the intestinal tract, will you get higher levels of resveratrol if you get into the bloodstream that way?
So,
and I had read some work that suggested that the sugar-substituted resveratrols can actually be transported across the intestinal tract, and I don't know then if they're hydrolyzed, etc.
yeah. So I think it just depends on whether they are subsequently hydrolyzed or not. And I don't know. I think those are all, I think really where it stands right now is, there's enough interest in resveratrol that there is big money right now in trying to find ways to increase bioavailability, and those kinds of things are being done. At the same time, I'm sure there's opportunities for things that aren't being done, ideas that haven't been explored. So those are all feasible.
So you haven't seen any literature on that subject?
But I haven't looked for that in particular. To be honest, most of our effort, this is one student doing this. This is only, this is something new to the lab, and it's one student's work. And most of the effort has been expended, and quite honestly, most of the interest is in exploring the molecular mechanisms of what's going on. Because, and again, what we do when we undergo an exercise like that is we identify other things. For example, this beta agonist, DPN, people have been playing with that in rats and getting all kinds of nice, beneficial cardiovascular and related effects in rats. So, there are alternatives to resveratrol as well. And this is not toxic, so this can be included in the diet as resveratrol would.
I might want to, somebody else?
Okay, no, last question.
One want to share with you an interesting story that I heard when I was attending a conference on lycopene in tomatoes. And Campbell Soup spent tons of money to try to prove that lycopene has beneficial effects in relation to a number of diseases like heart disease and so on and so forth. And at the end of that study, it spent millions and millions of dollars, and there was no effect that they could actually witness. And then someone came up with the idea that, oh God, we should have been making it as a tomato sauce, where there's a lot more fat, and so these chemicals end up in the oil. And under those conditions, you might see a beneficial effect. And of course, the problem then becomes, will the industry come back and say, well, we're going to invest even more money so you can prove the story that you're saying? And part of the problem is, A, overselling the story on the one hand, and industry not realizing how complex the experiment is to do to prove that a certain metabolite has these types of beneficial effects. That's why it's the epidemiological studies that are telling us these things are good for you. Now, how do we go from the epidemiology to illustrating the beneficial effect in a scientific manner, as you're trying to show?
Yeah, I think that's sort of happening, is that there's a lot of, there are some human trials now introducing resveratrol in different ways. Our particular discovery was linked to the mechanism. We think that manganese sod is in fact the mechanism. And I left out a bunch of data that shows us that it's not other pathways. Sirtuins, you're probably familiar with sirtuins and resveratrol in that story, and it's not. This is not. We've done the experiments with sirtuin knockout mouse cells, and we don't see that. We see nothing change, so we see all the same effects. So it's not a sirtuin story. So our contribution, really, the main contribution, is in showing what is happening. At the same time, there's a lot of money being invested in clinical trials, I guess, epidemiological studies to increase resveratrol delivery. But I think without knowing, without having a firm grasp of the science of what's really going on, and there probably are, I'm sure there's always bad and good effects of something like that, any hormone in the human body.
So it's a problem of taking pills with 10 grams of it. And if a little bit is good, more is even better.
Well, see, I mean, for example, I don't take resveratrol. It's premature. I mean, I've read enough stuff, and I've read enough stuff that one study contradicting another, of course, because it's at that stage in the evolution of the whole thing, that it's just too premature and lots of things.
I'm sure you're going to say except in wine, right?
Well, I mean, who knows? That's the. I think that this whole French fact so resveratrol was historically identified as, already they knew about the French fact. Red wine and high-fat diets somehow weren't negatively affecting the French, and maybe that's part of the story. You've got all of that. You do have a fairly high amount of resveratrol already in the wine, and you combine that with the fat, and that may be the perfect way to take it up.
And take up a bunch of other natural products.
And other things too, yeah.
That's what's really interesting. And it may be a much more complicated cocktail of chemicals that are getting into your bloodstream as a result of a higher fat diet.
Yeah. Well, the thing about this for anybody who wants to know, the thing about things that fat, everything that's in the gut that's getting absorbed, anything that's water soluble is going to go straight to the liver. It's absorbed into the blood cells, the blood vessels around the intestines and back here to the liver, except for fat. So fat actually makes this a partial circuit of the body before it comes back to the liver because it goes into the lymphatic system. So things that are inside fat are somewhat protected from that first pass of the liver, and that's where all the biomodification occurs. So that makes sense, that things that are hydrophobic or lipophilic want to be in fat, if they're packaged just so, they could get past that, and you could see an increased bioavailability. And along the same lines, where people have delivered resveratrol to mice and rats, and it's been labeled with radioisotopes so you can follow it, it seems to accumulate in brain. So that I've shown you a lot of brain data, and part of that was that the effects are always magnified in brain tissue. We don't really know why that is, but again, it's probably somehow related to the way it's floating around and being delivered.
Well, please join me in thanking